like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Welcome to the National University of Singapore Yong Loo School of Medicine Healthy Longevity webinars. Thank you for joining. I hope you're well. Professor Arfan Ikram is our guest tonight. I'm very much looking forward to a fantastic conversation. We have some roots in the Netherlands together, and you might have lots of questions. So please use the Q&A function and send us your comments, your questions for Professor Ikram. But we will start, as always, with a short presentation today by Stephen Dinish, a PhD student at our Center for Health Longevity at NUS. And he will talk about selenium and how that might mediate exercise-induced neurogenesis. And we will talk about the brain today, I can tell you. So Stephen, the floor is yours.
Thank you for the kind introduction. So today I'll be presenting to you some very exciting findings that was published in the journal Cell Metabolism very recently in March this year by Later et al. Selenium mediates exercise-induced adult neurogenesis and reverses learning defects induced by hippocampal injury and aging. Although the neurogenesis-enhancing effects of exercise have been extensively studied, the molecular mechanisms underlying this response remains unclear. In this study, the authors sought to identify mechanisms by which exercise regulates adult hippocampal neurogenesis. So as part of a previous study to identify proteins that are systemically released in response to exercise, the authors performed a proteomic screen on the blood plasma from exercised mice. Selenium protein P, also known as SEPP1, was one of the most significantly upregulated proteins present in the plasma of exercised mice, and were more than twice the levels of controls, as seen here in the figure on the right. But what is so special about this protein SEPP1? Well, SEPP1 is a selenium transport protein that supplies selenium to the brain and is known to be the most important for maintaining selenium levels in the brain itself. So having established that systemic SEPP1 levels are significantly increased following exercise, the authors investigated whether selenium treatment would also similarly result in an increase in neurogenesis. Selenium was infused directly into the hippocampus for 7 days, which resulted in a greater than a 3-fold increase in the number of proliferating cells in the dentate gyrus. The authors subsequently showed that this was due to the recruitment of quiescent neural precursor cells in vivo. To investigate whether effective selenium transport to the brain is required to regulate the exercise-induced increase in hippocampal neurogenesis, the authors generated SEPP1 knockout mice and subjected them to exercise for 7 days. Although they ran similar distances to the wild-type mice, exercise did not increase neural precursor cell proliferation in the SEPP1 knockouts as it did in the wild-type mice. Since selenium is a known antioxidant, the authors next investigated whether selenium could reduce the levels of intracellular ROS. Direct infusion of selenium into the hippocampus itself for 3 days resulted in the reduction of the total ROS content in these primary cells. However, there was no significant effect of selenium treatment on mitochondrial ROS levels. This indicates that ROS changes are due to a reduction in cellular ROS. The author subsequently went one step further to investigate whether selenium could rescue the age-associated decline in adult neurogenesis. Selenium delivery via the drinking water was sufficient to significantly raise plasma and brain selenium levels in both young and aged mice. Furthermore, selenium-treated aged mice had significantly more proliferating neural precursor cells and DCX-positive immature neurons in the dentate gyrus compared to controlled mice. Finally, to determine the effect of selenium supplementation on spatial learning and memory, aged mice given selenium were assessed using the active place avoidance task. In this task, mice are placed on a rotating platform and tested for their ability to learn to avoid a stationary shock zone. Selenium-treated mice were significantly better than the control mice at avoiding the shock zone, with fewer shocks received and a greater time to enter both the first and the second entrance into the zone. These results were corroborated using another learning and memory assessment test, also known as the Barnes maze. In this task, the mice were placed in the center of a brightly lit table with 32 evenly spaced holes. The mice were tested for their ability to locate one hole with an escape chamber. Similar to the active place avoidance test, selenium-treated mice had significantly improved Barnes maze performance and learned to enter the escape box faster than the control mice. So in summary, this paper shows that an upregulation in selenium transport promotes exercise-induced adult neurogenesis, and that mimicking this by dietary selenium supplementation can restore neurogenesis and even reverse the cognitive decline associated with aging. This novel mechanism could facilitate the discovery of novel therapeutic interventions which could be used to mimic the beneficial effects of exercise on cognitive function. So given that selenium is a cheap, readily available dietary supplement that is found in a number of commonly eaten foods, such as Brazil nuts, it could then easily be boosted in the diet of elderly people. This could be particularly relevant for the treatment of individuals who are unable to exercise due to advanced age, frailty, or disability. So that's all from me. Thank you.
Thank you so much, Stephen. I would say let's take selenium and exercise and test that in humans. And it might also be that the Rotterdam study has an answer to it, but I think we might want to discuss that in a second. Professor Arfan Ikram is professor and chair of epidemiology at the Erasmus University Medical Center, a big academic center in the Netherlands, and he is the principal investigator of the Rotterdam Study. And if you don't know the Rotterdam Study yet, it's really one of the international hallmark cohorts in the aging field, giving lots of answers how we age and why we age. And Professor Ikram also holds an adjunct position at the Harvard Chen School of Public Health in Boston. And he is really focusing on one aging aspect, and that's cognition. He is really interested in dementia and Alzheimer's disease and the relation with multimorbidity. He uses lots of data sets, and I think is really known for his epidemiological knowledge and looking at lots of new methodology like the causal interference methodology to understand the etiology and the natural cause of dementia. And excitingly, from August this year onwards, he will be the president of ZonMW. And ZonMW is the Dutch medical research organization, a very important organization for research here in the Netherlands, actually the most important organization. So we are very, very happy to have you here, Arfan, and giving your talk and your lecture about cognition and all the related topics. The floor is yours.
Thank you so much, Andrea. I'll just first start by sharing my slides. You should be able to see my slides now in the correct presentation view. It's a great honor for me to give this talk here today. And I have very fond memories actually of my visits to Singapore and to NUS and NUHS, I think already 7-8 years ago. Still have some ongoing collaboration. So it's it's great for me to actually give this presentation. Great honor, and also great to meet you again, Andrea. It's great to work with you and read up on some of the fantastic work that you and your team have been doing over the last many years. So today, as Andrea said, my field of application is dementia and Alzheimer's disease. At the same time, as an epidemiologist, I'm very much interested in thinking about methods and thinking about what we are actually doing with our research and what can we not deduce from our findings. So today I really want to take that angle and discuss applications and also methodological challenges of biomarker research, because biomarker research is one of the hallmarks of aging research, more so in dementia. And I actually became interested in thinking about such topics because dementia is, I would think, by far one of the most methodologically challenging diseases, given the fact that very often it is the last disease people suffer before they unfortunately die. So it comes with a host of challenges, ranging from competing risks to complex interactions, et cetera, et cetera. So that's kind of the angle through which I'll be giving my presentation today. Now, just to be complete, a few disclosures, particularly those that could be perceived as having some influence, although none of those is active at this moment. These are mostly consultancy I did in the last 3 years. So if you think about biomarker research, you see that there's a huge growth of biomarkers emerging in the field. And I really have depicted those here as layers, and people really talk about multi-layered omics research. Nowadays, any biomarker or group of biomarkers you can find, you add ohm to it and it becomes a whole field altogether. So you see here the exposome, epigenome, microbiome, metabolome, genome, etc. And the coming years, this plethora of biomarkers will increase even further. And really, this provides a wealth of opportunities for major discoveries, like we have seen with the GWAS studies, but also nowadays with the microbiome. At the same time, it also poses various challenges. And really, that's kind of the interface I'll be discussing today. Now, taking it to my own field, this is a diagram showing the current thinking on dementia and Alzheimer's disease and the origins. Basically, you see here on the x-axis the timeline from cognitively normal to dementia, and then the various biomarkers that have emerged in recent years. Now, note here that this paper is at 2020. The original model was conceptualized already, I think, 10, 12 years ago, and that consisted only of 3 lines. So really, you see here that as biomarkers start emerging, it really becomes important to identify their position in research and also think about how we are doing research. Now, if I would just look at biomarker research at large, I would really identify 3 categories in which we make use of biomarkers for research purposes. And I would actually dare say that ideally, we would keep these 3 categories separate. Very specifically, as I tell my students, really make a choice which one of these 3 categories you are performing your research in. Because if your ambition is to combine multiple of these 3 uses, the risk is that you start confounding and conflating some of these aims, and thereby making your research tougher for you to perform and tougher for others to understand. Now, the 3 areas for me would be really pure description, so really describing what is happening without any further conclusions. And then the second one is really identification. I think that's very often used for biomarkers, which is often called monitoring of disease or identification, risk stratification. We'll be talking about that in a moment. And then finally, you want to use biomarkers to really explain or understand the mechanism that's happening, because a biomarker, by definition, is a biological marker. It is marking something. So your aim is to understand that process that's being marked by this biological marker. And again, for me, these 3 aims really are separate because they force you to think in a different framework, thereby also influencing the analysis that you will do. And that is the main reason methodologically why I would say keep these three aims very clear and separate. Now I'll be going through all these three one by one, starting off with descriptive biomarkers or descriptive research for biomarkers. And really it is concerned with and designed only to describe the existing distribution of variables without regard to causal or other hypotheses. You simply just want to demonstrate what is happening. Now, if I'm talking about my own research in dementia, if I just want to know what is happening in the brain through the years or through the natural course of the dementia process, that would be a descriptive study. That's it. Typically, in an epidemiological sense, only descriptive measures are reported, such as prevalences, cumulative incidences, risks, incidence rates, or means. And that's where it stands. So the crucial point here is that you're not making a comparison across groups. Now, to demonstrate, this is a seminal paper in the dementia field already, I think 4 or 5 years ago, that basically demonstrated this hypothetical biomarker model I showed you 3 slides ago. This one actually was one of the very first in vivo studies providing real-world evidence in favor of that model. Now here, for instance, you again see the 3 groups: normal cognition, subjective complaints, and mild cognitive impairment. And you see, for instance, the percentage of amyloid positivity. Here you see other markers stratified by ApoE genotype, etc. and basically, these are just descriptions of these 3 groups. Now, you would be inclined to say, hey, I'd want to compare, let's say, this yellow-orange line to this blue one, but I think the authors did a very nice job of staying away from that. The aim was just purely description, and that's what they did here with these graphs. And really, this is really being used as a reference paper to actually compare other studies, like, hey, this is what's happening in the natural course, and let's see how our own study is comparing to that one. That's this first aim, description. Now, if I move forward to identification versus explanation, which are the second and third aim of biomarker research, as I see it, this distinction is important because whichever of the 2 you choose as your research aim, it will impact the study design and also the framework in which you will interpret your results. Now, interestingly, what you will notice is that both studies that aim for identification, and I'll go into that in a moment, as well as explanation, which I will discuss after that, all such studies, they present measures of association, such as relative risks, hazard ratios, odds ratios, mean differences. Both types of studies actually calculate this. But the crucial distinction is that the overall aim, so either identification or explanation, that determines whether these measures of association are then further analyzed for their predictive accuracy, or whether they are to be interpreted as causal parameters. And that's the crucial difference. And very often what you see is that studies present these measures of association, but don't take this next step. And for me, that is really, really crucial. Now, what does it actually mean in practice? So let's focus a bit on identification for a moment. Typically, the studies focus on identification or diagnostic studies or prognostic studies, in which you really want to use your biomarkers to identify persons that have the disease or are at increased risk of disease. Now, I'm really putting emphasis on the word identification because you're purely interested in knowing who has the increased risk, and not necessarily interested whether it is the biomarker causing that increased risk, yes or no. Now, once you have this, you can go further to use those measures of association and calculate these metrics, and everyone is familiar with these, but it's just this realization that if you really want to do diagnostic and prediction research, you really need to go on to calculate these metrics, of particularly the positive and negative predictive values, which are the clinically meaningful parameters rather than sensitivity and specificity. And then you go on to these 2x2 tables and these areas under the curve. I'm sure you're familiar with this, but this is really the pathway you take when choosing to do studies on identification. So either the diagnostic studies or the longitudinal counterpart thereof, which are the prognostic studies. Now, as I said a moment ago, for such studies, you actually do not need to adhere to methodological aspects that pertain to bias. So things like confounding, information bias, etc., because you're not trying to infer causality. You're merely interested in who has or does not have the disease. Compare, for instance, the situation of prostate cancer. The PSA, prostate-specific antigen, is a very, very strong biomarker for cancer. It can actually be used for screening purposes, but we know that PSA itself is not the cause of the cancer. It is merely marking that disease. A more extreme but very clear example would be that if I am interested in identifying people that have an increased risk of lung cancer, for instance, because I want to give general lifestyle advice to those people. If I'm interested in identifying people that have an increased risk of lung cancer, I can merely use carrying a lighter as a marker for increased risk. But we know that carrying the lighter itself is not the cause. But that's not the aim. The aim is not to infer causality. I'm purely interested in those that have increased risk, and carrying a lighter with you is a very, very strong predictor for lung cancer. Another example that I've borrowed from Professor Cecile Janssens, who's now working at Emory in Atlanta. She said that suppose you were doing a study that compared the chance of, so that was a study that is comparing men versus women in their chance of becoming the next pope, you would find hazard ratios or odds ratios of infinity, right? Because just think of a classical cohort study. You have men, you have women, you follow them up in time and see how many men and how many women became pope, and you divide the two risks and you get an exceedingly high risk ratio. So the measure of association is very strong, yet it is almost impossible to predict from the whole male population in the world who will be the next pope. So again, you see here the discrepancy between having a very strong measure of association does not necessarily translate into being a very strong predictor. And that's why you need to calculate those additional metrics. This is what I already mentioned here, that if your aim is merely identifying persons with disease or at increased risk, it doesn't matter whether that marker itself is or is not causally related, because that's not your aim. That's going to be aim number 3 if you're trying to explain or understand disease. Now, in this setting of diagnostic studies, also consider the reason for adjusting for covariates or confounders, because if you're not worried about bias, why are we adjusting for variables that may introduce confounding bias? So the reason for adjustments for covariates are different than what you would do in an etiological study. Now, examples of such prognostic or diagnostic models, there are many out there. I've just mentioned here a few very famous ones. The APGAR score obviously is one that everyone is familiar with. The framing of stroke risk profile for those working in neuroepidemiology, and I'm sure that other specialists from various medical fields recognize a few that I've mentioned here. Now, I'm just going to focus here on one that we developed for dementia. And really our aim here was identification, in this case, prediction of those that have increased risk of disease. And we basically made a prediction model, a basic model to be used in an outpatient setting, particularly family doctors, and one that was an extended model that included APOE, cognitive testing, brain imaging, etc. And here you could actually make the argument whether using cognitive testing or brain imaging as a predictor, whether that is causally interesting, because very likely these people already have the disease process ongoing. And that point is well taken. If our aim was causal research, we would not have used this one because it is already indicating that this person has the disease. But in case of prediction, and our aim was 10 years prediction, these are very interesting predictors because they actually help us to identify those persons that in 10 years time will receive the diagnosis of dementia. And we actually then perform calculated the metrics, in this case, the AUCs, areas under the curves of the various models. And you see here that they are exceeding 0.75 by and large, even 0.8 at instances, indicating that they really have clinical utility. Now, for me, the take-home message is that if we are now studying a new biomarker, let's say neurofilament light chain, tau, or any other marker that is emerging in the dementia field, and we want to test that marker for its predictive accuracy, we really need to realize that any new biomarker for prediction of dementia or AD will need to be tested against such existing models and evaluated for improvement beyond what is currently already achieved with the model that's there, and then also assess its cost effectiveness. And that's something that you sometimes don't see in biomarker research, because a new emerging biomarker is just tested by itself. And obviously it will then probably have a better prediction than a flip of a coin, but realize that for prediction research, it becomes an incremental activity. Now, a final word on prediction research or research on identification, because that's where I feel its position mainly lies, is this emerging field of artificial intelligence and deep learning, machine learning techniques. In essence, AI or deep learning is nothing else than a very, very, very sophisticated prediction algorithm, which means that is its position. So among the three aims I discussed, description, identification, and explanation, AI currently has shown its use in identification. You see here examples, which are not even the most recent examples. This field is growing very rapidly. What you will notice is that by and large, or perhaps almost all research showcasing the utility of AI is in the prediction area. Now, there are people out there, and there's a lot of commentaries on it, on this field that people call causal machine learning. I think that's an ambition that people have. I haven't seen it become a realization yet, and I think it may have some uses there, but I don't think AI will be as impactful in explanation as it has been in identification. It has some uses there, which I won't go into now, but for me, the positioning of AI is really in this realm of identification, diagnostic, and prognostic research. Now, if you go further with the third area of research, which is explanation, really you're looking at efficacy and etiology. Etiology, obviously, causal research, pathways, mechanisms, that's why you want to, that's how to explain the biological underpinnings of disease. And efficacy, obviously, if you want to intervene on something and really want to show that this intervention is the one that is helpful, you also looking at this explanation area. Now, obviously, the one that's kind of used as gold standard, especially for showing efficacy, are the randomized control trials, which indeed actually are trying to explain to you whether your intervention is the one that is having beneficial effect or not. Also realize that most of our basic sciences, so the wet lab studies, animal model studies, cellular studies, they fall within this realm. Basically, these are trials, but not human trials, but they are animal or wet lab trials. They should adhere to some of the principles I'll be discussing in a moment, basically the same principles that a randomized trial should adhere to. But I think by and large, most of the work that's being done on explanation research is not necessarily the trials or the experimental studies, but really the observational studies. So the cohort studies such as the Rotterdam Study and Framingham, but also many clinical studies are observational in nature. Now realize here that very often when designing such studies, in this case for explanation purposes, you very often do take an RCT as a hypothetical starting point to design the observational study for etiologic research. And for that, I will refer to the vast majority of causal inference literature that really helps you and guides you step by step how to take this hypothetical trial and translate it to your actual observational research. And within this realm, obviously the cohort studies are those that most people are familiar with, but we realize that case control studies and cross-sectional studies, if conducted properly, also have a position in explanatory research. They can provide you causal answers as long as you realize their limitations and also their strengths. Now, here, and this is just circling back to what I said earlier, similar to diagnostic and prognostic studies, explanatory studies also report on measures of association. So they too report on hazard ratios, odds ratios, relative risks, etc. The fundamental difference is that in explanatory research, we want to give these hazard ratios or these relative risks a causal interpretation, whereas in prediction research, we take those same measures and analyze them further for calculation of sensitivity, specificity, and all those metrics. In causal research, we stick with those measures of association, and we want to give them a causal interpretation. So instead of saying the relative risk is 2, we want to say that this exposure causally increases the risk of disease twofold. That's what we want to say. And for that, we need to make sure that in such studies, we take into account issues of accuracy, which are power issues, issues of external validity, and issues of internal validity, which are the biases. So for instance, the confounding bias, the information bias, and the selection bias. And once we have minimized the impact of some of these methodological issues, that then leads to a causal inference. And that's why in such settings, if we are convinced that we sufficiently took some of these methodological challenges into account, we can then interpret this relative risk of 2 as a causal relative risk. Now, it does bring some other challenges, especially if you're doing biomarker research and how to look at that. Because you need to understand that the biomarker very often itself is perhaps not the molecule of interest. It is actually marking an underlying disease process. So let's say if I'm interested in amyloid in blood, obviously I'm not so much interested in the amyloid in blood for Alzheimer's disease, but I think that amyloid in blood is reflecting the disease process as it is happening in the brain. This holds for many other molecules. So if we are interested in a certain outcome, basically we are interested in this arrow. We actually want to know whether that underlying disease process causes the outcome. Think about inflammation, atherosclerosis, like if you take calcification as a proxy, as a marker for atherosclerosis, again, it's a biomarker, it's not the disease process itself. But instead of this arrow, which we fundamentally cannot quantify, we instead quantify the association of the biomarker with the outcome. That's not necessarily a bad thing. It's just that you should realize that this is the underlying association that we are interested in. Now, very often you see here that the biomarker and the underlying disease process are not 100% correlated. Like, CRP is used as a biomarker for inflammation, but CRP does not itself reflect the whole vast inflammatory process. It has some correlation. What is interesting here is that if you see this, and we are looking at this association of the biomarker with the outcome, basically it means, looking at this diagram, that association is fundamentally confounded by the actual disease process. Now, I'm sure many of you hearing the word confounded think like, oh, that's a bad thing. But in this case, it's not a bad thing. It's just a realization, like hey, this diagram is the one that we use for confounding, because the biomarker outcome associations is confounded by the underlying disease process. Nothing wrong there. Just realize again that that's inherent to the fact that it's a biomarker. It does become problematic in a situation in which you are, for instance, interested in some other exposure of interest. So let's say we are interested in glucose levels and dementia, and you want to adjust for inflammation. Now, in that case, we would use a biomarker for inflammation because we cannot measure inflammation itself directly. And because that biomarker is not perfectly correlated with the underlying disease process, it becomes a fairly weak proxy. Now, in this situation in which you are interested in this, in this arrow, and I hope you can see my mouse moving over the screen. So in this situation where we are interested in the exposure of interest to outcome, and we adjust for biomarker, there we know for sure that using a biomarker as covariate, intending to thereby adjust for the underlying disease process, that will always lead to residual confounding, because that biomarker is not fully capturing the process that we wanted to adjust for. And that is a bad thing that you cannot do anything about, but at least something to mention in the limitations, for instance, like, hey, we adjusted for this or that, but these are not optimal, perhaps even weak proxy confounders that we adjusted for, and it may have led to residual confounding. Now, I just want to refer finally to two papers that for me really were very helpful in shaping thought process and also form some of the bases I discussed here. This is a recent paper by the group in Leiden that really deals with the same topic, prediction or causality, and they did a scoping review, so not a systematic review, but just a scoping review to get a sense of what's happening out there, to what extent studies in very often major clinical journals really separate out their aims as being either prediction or causality. And what you see here is that there were studies focused on etiology, there were studies focused on prediction across the various fields. Luckily, there weren't many studies that at the outset said, we want to study both in one. But I think the take-home message here is that about 1 in 4 studies actually conflated the aims. So they perhaps introduced the study as a study focused on prediction, but then started interpreting the findings in a causal framework and vice versa. And that's something we should realize to avoid, because it can lead to confusing and sometimes often conflicting results. This is the second paper that I wrote myself with a colleague of mine, again on the same topic of so instead of calling it prediction and causality, we call it identification and explanation, and add a third one, which is description. And again here, comparing side by side, what is the overall aim of these types of studies, what statistical aspect is of interest. So here we really focus on fitting the model and evaluating the model as a whole, whereas here in explanation studies, we are purely interested in the measure of association, etc., etc. So concluding remarks, I think biomarkers with all those omics layers emerging hold great promise for groundbreaking new research opportunities. At the same time, we need to be aware that we position those biomarkers, and the same biomarker could be used for any of these 3 research aims as long as we realize what it is being used for: description, identification, or explanation. And this distinction is important because this will then determine your study design, your analysis, and most importantly, the interpretation of your findings. And with that, I want to stop here and thank you for your attention.
Thank you so much, Arfan. That was a great lecture, how to do proper research, I would say. And I very often have the discussions with my students too, and ask them, what is the difference between an association and a correlation? And what do you want to do? Do you want to describe, or do you want to identify? So rather do risk stratification and risk prediction. Let me ask you first a very broad question, because I think you also, as reviewer of articles and editor, we often, I think, find papers with very nice content, but not using the right epidemiological analysis and the strategy. So the method section could be rewritten, or the analysis should be redone. How do we improve that? And especially as clinician, I very often see that MDs do epidemiological studies. How do we improve it? How would we change it?
I think it starts off with awareness, right? So I hope my lecture contributed to that. It's really awareness that people should understand what they're doing research for, and what's happening in the research. And one thing I've often said also to funding agencies, for instance, as a reviewer, is what would you rather have? would you rather have extremely important research being done in a bad way, or would you like to have research that perhaps is not directly clinically impactful, but it's at least being done properly? And I think the first scenario is a very dangerous scenario because lives could be lost. Think about the whole situation surrounding COVID in the last 2 years. There was a vast emergence of science, which in my opinion was not of the highest standard, and what kind of impact it may have had if some of those findings, because the topic itself was extremely important, and if those findings would have been taken forward, and for some, I think actually they were taken forward. So I think that's kind of the balance. It starts off with realization, and then it's realization, but also then training yourself in some of these methods. But I can also fully well imagine that one person cannot do everything by him or herself, and that's why we nowadays have team science. So make sure that you do have a proper methodologist on board for any research that you do conduct.
Yeah, so more education for students, but also for researchers in general, I would say. Yeah. Okay, so but on the other hand side, so we have sometimes the lack of the quality of the methodology. On the other hand side, we also have such a hype to publish and to do all the analysis, and I think have so many prediction tools in the end. If we stick to your second topic, that's the identification risk stratification. And what I see is that not many of the risk prediction tools, even if they have a quite clinical good predictability of a 0.8 or something, they are never reaching clinical practice. And so what is your thought about that? Because as a clinician, I think I use 10 tools, but that's it.
Yeah, I fully realize the difficulties that you're facing here. Very, very often what you see is, and that happens in every field, like, in the field of genetics before the GWAS studies, every lab had its own favorite SNP that it was studying for 30 years. Every lab has its own favorite brain region that it's studying for 20 years. Every lab has its own favorite biomarker, and every lab has its own favorite prediction rule, which they think is the best. So what you happen is that there's no crosstalk between such prediction rules. Now, having said that, I am aware of a few recent developments in which various studies are being pooled and trying to create one overall prediction rule. So I think that's a good thing. At the same time, also realize that creating good prediction rules is really difficult. Now, just looking at, for instance, the Framingham scores, either for myocardial infarction or stroke risk, they are very simple rules, like asking about someone's smoking, taking a blood pressure, cholesterol, lipid levels, gender, and I think that's about it. And they already have a very, very strong and good prediction. Now, you need to realize that if I would have some fancy biomarker, or let's say a genetic score, which are nowadays also very, very, very hot, if I just compare such genetic score against age and sex, obviously it will have good prediction. But if I would compare it against the Framingham score, the incremental value becomes very, very limited. In fact, the other day I was discussing with someone, and we were actually amazed that the Framingham scores were developed in the early 90s, I think 1990, 1991 with Full Wolf and a few others. And it took 30 years to get a small incremental improvement by adding either anti-pro-BNP or adding calcification, for which you need to do CT scanning and everything. So I understand your point, but it is just extremely difficult to get good prediction rules, or actually improve good prediction rules. Now, that's where I think AI has shown its purpose so far, because that really leads to meaningful improvements all the time.
Yeah. And I think the second thing is that cohorts or the PIs of cohorts have to work together to do all the external validations of these prediction tools, and to also make sure that we have a gender balance, ethnicity balance, etc. Because otherwise we end up which is happening very often at the moment in the geriatric field, that having tools for the individual with hypertension and diabetes, but not the one with the Parkinson's disease.
Exactly, and that's again with prediction rules, but also for research in general. To what extent do your findings in one setting also are applicable to another setting? And if not, you need to redo the research in that other setting as well. And that is prohibitively limited to do all studies in all different backgrounds.
Yeah. May I ask you a little bit, I would say, a provocative question? I know that you're really focusing on the Alzheimer's and dementia field. How ethical is it to build all the prediction tools without having any therapies, and especially already knowing 15 years before you have the disease that it's very likely with a certain chance that somebody has something, a disease, and carrying that with them? So where is the field, and what's your advice?
So this is a question we thought of ourselves as well. Like, hey, even before embarking on developing our model, what does it mean, and can it mean anything? But we still went ahead, on the one hand, because of academic interest. On the other hand, because we considered that as we speak, many prevention trials are ongoing. So at the very least, what you could do is inform the trials like, hey, these are the individuals with the highest risk that you should be preferably recruiting into your trial because they will benefit most from any prevention. Now, that's how we started off. And I think that purpose was served. But once we had our findings, actually one of the family doctors pointed out to us the following, which was, and I didn't show the data here, is that we made this chart just like you have the charts using by GPs. You know, we have the green color, yellow color, red color, like where you end up. We made this chart for dementia. And what we found is that people that had the worst, quote unquote, profile merely had a 10-year risk of about 25 to 30%. So it was good prediction, but 20-30%, is that sufficiently large to intervene on something for that person? If you tell someone, hey, you have a 10-year risk of dementia of 25%, should you not intervene on that? So that was kind of the discussion we were having. And that GP pointed out to us, why are you focused on those persons that have the worst risk profile? Because the risk is only 25-30%, and you very, very likely won't do anything because it's a 10-year risk. But look at the other spectrum, people that have the most optimal profile. And in our case, it meant someone of 60 years old, did not have memory complaints, did not have had a stroke, and was still independent in doing all their IDL activities. Such an individual only had a 10-year risk of 2%. Conversely, it meant that this person, for 98%, was sure that he or she was not going to develop dementia. And the GP said, that's the use of the model at this moment. You can reassure persons that they are not at increased risk. So don't look, because this model will not help you for those that have the highest risk. But most people coming into my practice come to me telling, doctor, I'm worried because my father or mother got diagnosed with dementia. He or she is 90, 95 years. I'm 60, 65, and I'm worried about my risk. And in this case, you could use this prediction rule and say, well, based on the prediction rule, I can reassure you that for the next 10 years, your risk of dementia is exceedingly small. So don't worry about it, for now at least. And I think that's the value that these prediction models for dementia may have at this moment.
Yeah, I get that, but hopefully that person just with 1 or 2% of risk will still be physically active after you told.
I hope. Yeah, the reassurance shouldn't mean that don't do anything anymore, but means at the very least continue in what you've been doing.
Yeah, because all the preventative studies you're also referring to, lots are with lifestyle, as we also do the SINGER study in Singapore. So so there's always a downside. Before going to Kamal Pavis, who has, I think, lots of questions from the audience, I would like to ask you one more question. So you started with this beautiful slide of all the omics data. And indeed, lots of groups are focusing on one or the other. Not many are combining them. I know you have lots of these data. So, and I know AI might have a very good good role in there. But how much of the omics, in your opinion, do we really need? And aren't we measuring, especially if we want to talk about risk prediction, aren't we measuring lots of the same, but only upstream or downstream of certain mechanisms?
Yeah, you're absolutely right. I think for prediction purposes, it depends on how accurate do you want to have your prediction, and secondly, what can you offer the patient for which you have predicted the risk. And I think for prediction purposes, especially with the strength of AI, yes, I don't think we need, for the time being at least, many more omics layers. I think if we can digest the omics layers we have now and get some of prediction rules, that's already fine. It becomes a much more tricky question if we say, what about omics in the field of explaining and understanding disease, hopefully thereby identifying a modifiable target or druggable target? That's a difficult question because 15 years ago, when the genome was kind of unlocked through GWAS, people thought, well, this is the end of the road. Here we are. We have it. Now, 15 years later, we actually got even more difficult and complex questions to answer than we had 15 years ago. So, yeah, I think for causality, it's the jury is still out. Of course, there are examples there where through GWAS studies, a druggable target has been identified, or certain subgroup of cancer has been identified for which you can do gene therapy. But for the bulk of diseases, if I just take it to my field of dementia, there still isn't anything there because, yes, a lot of progress has been made for these familial subtypes, typically autosomal dominant mutations for Alzheimer's disease. And even there's not a clear treatment even there yet, let alone this for the sporadic form. So, yes, I think these omics layers are very exciting. At this moment, they remain academically exciting and not so much clinically exciting, but hopefully that will change in the next 5 to 10 years.
Yeah, fully agree. Very exciting. Camille, what are the questions from the audience?
Thank you for the great talk, Professor Irfan. We'll be taking now some questions from the audience. There's an interesting one. So, given your critique of the use and limitations of biomarkers, what do you think about FDA's accelerated approval process, which gives preliminary approval based on biomarkers without clear efficacy?
Yeah, that's a difficult discussion because let's say the viewpoint I brought here was, and I will admit, a purely methodological viewpoint, like, hey, this is how it should look like in an ideal world. At the same time, you know that in the real world, there are other constraints, other urgencies, like we have seen with COVID. Sometimes a decision has to be taken. You simply cannot say, let's wait 6 months for the ideal study to be conducted and see what comes out of that. So that's kind of a balance that needs to be found. And I'm kind of happy that I'm not the one that has to take that decision, but I fully understand that someone has to take a decision there. And I don't think that's a bad thing, as long as you realize under what circumstances the decision was taken, and if you're open to revoking or developing that decision further as more and more evidence becomes available.
Another question is, have you identified any other potential biomarkers for other dementias from the Rotterdam study, like for frontotemporal dementia, vascular, or Parkinson's disease?
Yeah, so that's an area of active research. And what you see, the picture that is emerging is that, yes, these subtypes of dementia are different entities. At the same time, what you see is that they also have a common biological trunk of shared risk factors, shared pathological processes, which for me is perhaps even more exciting. Like, hey, what are the similarities of all these different dementia types? Now, the question is, somewhere they diverge. So they may have a common trunk of leading to brain pathology, and somewhere they diverge into the various entities we have. Now, in the Rotterdam study, we try to be at the forefront of technology. At the same time, we're at the forefront of technology that is ready for high throughput application. And I think that means that we are more or less limited to plasma biomarkers for our study. And that's where you see that, and neurofilament light chain, tau levels with the various species thereof, glial biomarkers are the ones that are of interest and are emerging. I am aware that in some of the more clinical or fundamental studies where you have direct access to brain tissue or CSF, more exploratory and experimental biomarkers are being studied.
All right. And related to this, one person asked, is hearing impairment associated with cognitive decline or dementias?
That's a controversial one. I know that the Lancet Commission mentions it as one of the 12 risk factors, along, for instance, with depression that has the same issue. This really ends up in what I discussed about explanation. Do we have sufficient evidence, mostly from observational studies, because obviously you cannot do an intervention forcing people to become deaf, for instance, and see what it does with the dementia risk. So it ends up being a question of observational research and to what extent we feel that observational evidence is sufficient for us to draw causal conclusions. And I personally feel that it could go either way. It could go either way at the same time, because we also know that an ongoing dementia process, because we know dementia is a diffuse disease in the brain, perhaps it starts in one location, but it then spreads throughout the whole brain. It will also affect the auditory cortices and the auditory neurons. So sooner or later, your hearing will go down, will deteriorate due to the dementia process. And then the question becomes, was it the hearing loss that then subsequently led to clinical dementia, or was it the reverse causality? I think, and for many of these dementia risk factors, that is still a question, especially so because it's a disease that has, a long preclinical phase, and the formal diagnosis of dementia is merely one dot in this whole spectrum, which is in contrast to a stroke, for instance, where you can really say this was perhaps not the timing, but at least this was the day the stroke occurred. For dementia, you cannot state something that easily, because very often by the time the diagnosis has been made, we know that the earliest signs in the brain for dementia were already present at least 10 years before, perhaps even 15 to 20 years. And that makes it methodologically challenging to draw definite causal conclusions.
Maybe another one would be, what are practical recommendations one can do to avoid dementia? That's what usually people want to know when they attend such a talk, I think.
So what you can do practically at this moment for dementia is, and for that, I really look at our colleagues from the cardiovascular field, is basically I say what is good for your heart is good for your brain. So it does mean that things like, controlling blood pressure, sugar levels, lipid levels, etc., that all is not only good for your heart, smoking, of course, as well, but also for your brain. So you should avoid smoking and maintain your cardiovascular profile. I think the good thing about the brain is that you also have a few additional things to work on, which we very often put under the umbrella of cognitive reserve or cognitive resilience. Think about social activities, cognitive activities, psychosocial health, because I'll compare it to physical exercise and your muscles. If you want to keep your muscles strong, you need to regularly exercise, because otherwise they weaken. Similarly, I think the same is happening with your brain. It doesn't mean, and that's the second follow-up question I often get, is that, I have someone who is demented. If that person starts making puzzles, will that person recover? The answer to that is no. So for me, I think the conclusion is that brain training can help you maintain brain functions that are still intact. So it can prolong the period that you are still capable of doing those activities. But once you lose a certain brain function, then brain training will not retrieve it anymore.
Last question, Camille. I think this really also nicely highlighted how important aging and the aging process is also for the brain. But I think we will leave that to another session. But Camille, one more question.
One of mine is, how early do you think we should start treating in studies? Do you think studies these days are too short?
I think the studies nowadays are not necessarily too short, but they are just constrained with the time they have. I mean, running a clinical trial for 5 years really is a very long period, especially considering the fact that you want to have participants compliant with your intervention. You want your participants to keep coming back to your trial, because loss of follow-up can be finessed to any study. So yes, an ideal trial would last 20 years, but it also means that if I start a trial today, I will only get my answer in 20 years time from now. Will it still be relevant? So yeah, that's kind of the balance you need to strike, because it by definition means that the trials that are providing their results today are those that started typically 5-6 years ago, if I'm talking about prevention trials. And how relevant are some of those interventions after 5 years, 5-6 years still? So it becomes a balance. There's no gold standard or a definite answer. And I think that's what I like about epidemiology, is that of all the various study designs you have, each one has its own strengths and merits and downsides. And in the end, you need to really synthesize your evidence from various sources and various study designs to get the answer you're seeking. You won't have one study design that is providing you the full answer to all the questions and sub-questions you may have.
That leads a little bit also to the congratulations of the Rotterdam study having so much follow-up and not much lost of follow-up, as I know of. So very well done. So it's really one of the hallmark studies cohorts. Thank you so much, Professor Ikram. I'm hopefully seeing you again, Arfan, at certain times in the Netherlands. Thank you so much also for this great talk, and so much good luck for your new career step. Hopefully, and Camille also, of course, thank you for providing all the questions. Very important, leave your, in the chat function, the panelists and all attendees option, your comments and the feedback. And we have some very great news after the end credits, after this talk. We invest heavily in social media. We have a Twitter account, a LinkedIn account now. So please visit us and we will continue the discussion. We also have a new website. We showed some of the pictures in the beginning. Also, please look at us, our website, and find out about our mission and vision and the studies we are doing. And if you would like to participate, please do so. There will be the links in the chat box and QR codes. Then, of course, there will be a next session, and that's the 16th of June next week. I will be the next host, and with me will be Professor Michael Risto. I will leave you, as always, with a video, and I would say in the video we shouldn't ask, are you still driving, but are you driving? Because I think that's a much better and relevant question. Take care.
I can't even believe that I'm sitting talking to you, and the energy that you have at 105. I mean, I couldn't believe when they told me I was going to meet somebody 105, and that you're driving still?
Yeah, I got my license renewed this morning, and I passed.
She's never had.
I just found out that I'm the oldest living driver,
yes,
in California and maybe in the United States.
I would think so. Yes, I would think everywhere. So, and you've never had a ticket. You've never had an accident.
That's right.
Completely. And you're just so vibrant. And I guess everyone must ask you this. But what's your secret? What do you do that just keeps you so alert and aware?
I do have a secret.
What is it?
If I tell it, then it won't be a secret.
Yeah, that's true. Well, I would think having a sense of humor is the secret.
Well, I would say I've always been very happy. I've had a happy marriage. I've got a wonderful family. I think positive.
That's really important.
If something happens, I can't do anything about it, so I don't let it bother me.
I'm feeling like sunshine, like springtime. Like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.